Thank you, Dr. Mitra, and also Dr. Rittenhouse, for this opportunity and also for organizing a really outstanding session. It's really been exciting. Like other speakers here today, I'm also an engineer by training, and my laboratory is interested in the development of molecularly targeted imaging and therapeutic agents for retinal disease. And today I'm going to talk about our efforts towards targeted therapy of retinal vascular diseases, primarily wet AMD, but also a few applications in proliferative diabetic retinopathy. Like many in the audience and many of the speakers, we appreciate that engineering imaging agents and drugs on the nanoscale, which is the scale on which many critical biological interactions occur, can enable very novel and powerful clinical applications using some of these devices you see below. And so our goal is to harness nanotechnology and nanoengineering for applications in diagnosis and therapy of retinal diseases. Pathologic ocular neovascularization is a common component of many blinding diseases, including diabetic retinopathy, which features the pre-retinal vitreoretinal neovascularization outlined in letter D, or also the subretinal neovascularization, like the type observed in neovascular or wet AMD, which is outlined in letter B. And what we want to do is to engineer nanoparticles that can sense dysfunctional endothelial cells participating in ocular angiogenesis, which spare healthy tissue, and that has very important implications for how we treat this disease and manage it. While I should say that, while anti-VEGF therapies have truly revolutionized the practice of ophthalmology as we know it, many of these patients with AMD and increasingly DR, despite timely therapeutic interventions, are nevertheless still going on. A large fraction of them are still experiencing progression to legal blindness. So I think in the field, clearly some other therapeutic options are warranted, either as an adjuvant to anti-VEGF therapy or maybe even an alternative. And I think a lot of clinicians and pharmaceutical companies, as Dr. Rittenhouse elegantly outlined in her talk, are very much interested in translating siRNA to the clinic for ophthalmic applications. As you know from the previous speaker, siRNA encodes sequence specificity, and which can be used to target virtually any intracellular molecule, including those that are currently undruggable. So siRNA gives us a very useful therapeutic avenue to targeting new targets as they're identified on the bench. And also, the chemistries of siRNA were outlined, which have improved their stability and specificity, and those advances continue to facilitate siRNA preclinical and hopefully clinical applications. And unfortunately, though, the siRNA delivery problem is not a trivial one. siRNAs, despite great advances in backbone chemistry, still can be compromised by poor stability in vivo, depending on how they're administered. There's always the potential for off-target effects on tissue, so we really don't want to target siRNAs or any RNA-directed therapy to healthy cells if we can avoid it. And therefore, this is really a promising therapeutic strategy, probably in need of an effective delivery vehicle. And so what we're trying to do is to use nanotechnology and nanoengineering and what we've learned from that emerging area of science to overcome these barriers to effective siRNA applications in the clinic. So, ideally, we would want to develop these targeted nanoparticles which protect the siRNA payload in vivo. Maybe it could even be administered intravenously to avoid the complications of intraocular administration. And ideally, these sentinel nanoparticles would home in the vasculature and seek out dysfunctional endothelial cells susceptible to preretinal or subretinal neovascularization, and even more so, participate in site-specific binding and triggered release of siRNA within these dysfunctional endothelial cells. And so we began a search, along with many other groups throughout the country, For molecular signatures of these neovascular lesions like you see in B and D, what do these endothelial cells feature on their surface that might be exploited for site-specific triggered release of siRNA? And we, as well as several other groups, including the Scott Cousins group at Duke, have identified VCAM1, vascular cell adhesion molecule 1, as a promising cell surface targeting biomarker on endothelial cells for subretinal neovascularization. So that's the choroidal endothelial cells. In experimental cell culture models, we can stimulate choroidal endothelial cells with an inflammatory stimulus, TNF-alpha, and ELISA shows an increase in VCAM1 protein. In addition, in an experimental animal model of choroidal neovascularization of the type that occurs in wet AMD, we were able to see focal upregulation of VCAM1 only within the laser-induced CNV region and not in the proximal healthy retinal tissue, which makes it perhaps a promising target. In addition, we've taken the RNA out of these experimental LCNV models, and we've measured an increase in VCAM1 mRNA expression as well. Just to explore the targeting properties of a VCAM1 targeted nanoparticle, we conjugated anti-VCAM1 peptides or antibodies raised in our laboratory to fluorescent nanoparticles, and we injected them intravenously in these mouse LCNV models. And we observed that VCAM1 targeted nanoparticles, but not a non-specific IgG targeted nanoparticle, accumulate nicely within the laser-induced CNV lesions. So this gave us some evidence that VCAM1 might be exploited for targeting, but up till now we've only really explored binding. We haven't really looked at internalization. But to our surprise, VCAM1 loaded nanocarriers featuring dye-labeled siRNA were nicely internalized within human choroidal endothelial cells within an hour. IgG conjugated nanoparticles were not. So knowing that VCAM1 could be exploited for both binding and internalization led us to design a polymeric nanocarrier approach for targeted and triggered release of siRNA. In this approach, we typically take a cationic polymer, and we've experimented with several and had varying degrees of success with several, but protamine and PEI are the most promising. These cationic polymers condense the siRNA, which we limit to 19 nucleotides for the length reasons that Kay was pointing out. On this cationic polymer, we shield that positive charge with a polyethylene glycol linker, which is acid cleavable. And on the distal end of that PEG, we can conjugate a targeting fragment, either a VCAM1 antibody fragment or a peptide. We hypothesized that this nanoparticle would be intravenously injected, bind to the subretinal neovascular lesions, bind to that cell surface VCAM1 on the choroidal neovascular lesion, get internalized, and we were hoping that that PEG, which is acid cleavable in the endolysosomal compartment, would be cleaved, thus re-exposing PEIs or protamines positive cationic charge, enabling release of the siRNA within the cytoplasm. We did a lot of quality control experiments. First, we wanted to make sure that the nanocarriers adequately condense and protect the siRNA. We also experimented with some anti-microRNA cargoes. And the polymers condense these cargoes and form nice monodispersed nanoparticles as measured by dynamic light scattering. Also using a DNA intercalating dye, an ethidium bromide dye exclusion assay to examine protection of the siRNA. We incubated the nanocarriers in a cocktail of nucleases and showed that the complete nanocarrier formulation, but not just the conventional naked siRNA, were protected from degradation effectively, which would be important for in vivo applications. We also performed zeta potential analysis. These particles are negative, which is important in vivo for reducing nonspecific binding interactions and promoting only specific interactions with that cell surface VCAM. So, and that's regardless of whether the carriers have siRNA or microRNA. Using a FACS assay, where we stimulated human choroidal endothelial cells with TNF-alpha or left them untreated, we were able to show that nanocarriers are capable of antibody-specific interactions with dysfunctional endothelial cells. Only the VCAM1 coded nanocarriers were capable of binding to the TNF-alpha cells, not the isotype control, nonspecific IgG-coded nanocarriers. Of course, if we take the PEG and the antibody off, we just have a lipofectamine type transfection reagent which goes into cells and is insensitive. Using RT-PCR on in vitro with human choroidal endothelial cells, we were able to confirm that VEGFR2, this was the target we were targeting with our VEGFR2 siRNA cargo. We observed through PCR that nanocarriers were capable of sequence and antibody-specific inhibition of VEGFR2 mRNA expression. So only nanocarriers with the VCAM1 targeting antibody and the VEGFR2 siRNA combined were capable of knockdown, not the IgG-coded nanocarriers or VCAM1-coded nanocarriers with a nonspecific scrambled siRNA. When we injected these nanocarriers targeting VCAM1 in mouse models of LCNV, we were able to observe that indeed the nanocarriers home to focal sites of experimentally induced CNV. Green is ICAM2 that outlines the endothelial cells of the LCNV lesion, and we can see that in many cases the nanocarrier colocalizes with those choroidal endothelial cells. And in day 5, you can see that the nanocarriers kind of also localize around the lesion, which is now leading us to look at the underlying RPE proximal to the lesion to see if those express VCAM, because RPE can, under certain conditions, express VCAM. So that would open up some novel applications for targeting VEGF expression in RPE, which would be complementary to this therapy targeting VEGFR2. Notice that a control did not accumulate appreciably within those CNV lesions. In addition, we measured the CNV lesion volume at day 5 post-laser injury. The nanoparticles were injected on day 1. We observed, again, analogous to the in vitro results, antibody-specific and cargo-specific inhibition of choroidal neovascularization. Only the VEGFR2 siRNA nanocarriers functionalized with VCAM1 were capable of inhibition. Also, we co-injected in separate experiments a toll-like receptor 3 antibody. Kay mentioned that there are TLR3 non-specific therapeutic actions of siRNA, and by co-injecting this neutralizing antibody and getting the same result, we were able to begin to demonstrate that this effect is TLR3 independent. In addition, we've performed a variety of toxicity assays to demonstrate that in vitro, these nanocarriers are well tolerated by the choroidal endothelial cells. We're still taking care of ERG and tunnel assays to confirm this in vivo. We've swapped the cargo out for the siRNA with the anti-microRNA and observed similar CNV inhibition. We're also now trying to target pre-retinal neovascularization with either endoglin-targeted nanocarriers, so we swapped the VCAM1 for the endoglin antibody, or the ICAM1 nanocarriers, where we swapped the VCAM1 for ICAM1 targeting nanocarriers. So we also have targets that work well for proliferative diabetic retinopathy. The same platform can be used to inhibit pre-retinal neovascularization as well as the wet AMD subretinal neovascularization. So we believe we have an approach that begins to address some of the key barriers that are kind of hurdling, that constitute a hurdle for siRNA clinical applications. We believe we have a modular design. You can swap antibodies, you can swap cargoes, and hopefully use that, exploit that property to target multiple targets for more potent inhibition of neovascularization or regression of existing neovascularization. So I'd like to acknowledge my coworkers and my collaborators and funding sources without which this work would not be possible. And thank you for your attention.